So yeah, if you could do that for us, just introduce yourselves a little bit.
Sure, maybe I can go first. So my name is Michael Köpke. I've always been very fascinated about biology, and that eventually led me to pursue a PhD in molecular biology and biotechnology at the University of Ulm in Germany. And for the last 13 years, I've been working for a startup called LensaTech that essentially treats waste as an opportunity rather than a liability. I started with the company in New Zealand with maybe a team of perhaps 30 people at that time. I think now we're kind of over 250 people and headquartered in Chicago. So I think it was really exciting to see that growth of the company. And my current role in the company is I'm the VP of synthetic biology. So I lead the strain engineering R&D program that LensaTech has.
Great, thank you.
Yeah, thanks, Michael. That's a great intro. I'm Mike Jewitt. I'm a professor in chemical and biological engineering at Northwestern University, where I also direct the Center for Synthetic Biology. Really appreciate you guys having us today. You know, at a higher level, we've been collaborating with LensaTech for several years now, really trying to think about how can we advance our capacity to partner with biology to make products that we need every day in a sustainable and renewable way. And so this project was a particularly fun and exciting one, and glad to be talking about it.
Great, thank you. Yeah, so we invited you after reading your paper on the carbon negative production of acetone and isopropanol. And yeah, we just wanted to ask you some questions and just get a, I guess, overview for, I thought it'd be a very interesting topic for our podcast, because I think we found it very interesting and had some questions to ask. So I think a good place to start maybe would be to sort of describe what exactly is carbon negative and sort of like the circular economy. What do those two terms actually mean?
No, I think this is a really good start with. So I think I mean, we obviously kind of have a big issue at hand with the climate crisis, and I think we need to kind of reduce our CO2 emissions, and even I think pull out CO2 out of the atmosphere. So if you look at for example, energy production, I think there's ways to do that carbon free. I think with wind, solar, there's a lot of renewable options. But there's a couple sectors which are really hard to decarbonize. I think one is the chemical industry, which I think has several gigatons of CO2 emissions a year. And likewise, I think there's the heavy industry, for example, steel making, cement production, that things that we need, but I think again also producing several gigatons of CO2 emissions each year. And chemical production of the hydrocarbons always need to have carbons. I think we'd be very mindful about how precious our carbon is and try to recycle that carbon. So I think in the paper, I think we've developed new routes to make acetone and isopropanol. So today, those routes are all produced from chemicals are all produced from fresh fossil, from oil or natural gas. And this production process, the cumin process, is for one, it's relatively environmental unfriendly. I think has a lot of toxic waste. But it also, per kilogram of acetone you produce, I think you have 2.5 kilograms of CO2 going out in the atmosphere. So our process now allows to make the same chemicals, but use essentially waste gases as input. So that can be steel mill emissions or CO2. And by capturing those waste gases and essentially locking those into the product, we can essentially remove CO2 from the atmosphere. So for acetone, for example, we can have a negative carbon footprint. So that means we can have minus 1.79 kilograms of CO2 per kilogram of acetone produced. So that means the 1.79 kilogram of CO2 are then locked into that acetone molecule per kilogram of acetone produced.
That's cool.
And acetone is a building block, and for example, for plexiglass, so essentially it's locked in and therefore forever.
Yeah, that's great. I mean, that's just reading that and hearing about that is just like, it's things that you sort of think about and then never imagine that possible. But then, you know, people like you are doing it, and it's great. I was wondering, you mentioned that the chemical industry in particular is sort of like, it's difficult to decarbonize. Like, what did you mean by that? Like, what were the issues that you've been facing when it came to realizing this I guess process?
Yeah, so I think obviously, I think for energy, as I said, this can be carbon free, but I think our chemicals and our everyday products are all hydrocarbons. So I think they need carbon. So I think the question is then, where does that carbon come from? I think, and right now, I think pretty much everything is coming from fresh fossil carbons. I think it's really kind of important that we consider using waste carbon. And then I think with how far we are now, I think it's important that we even kind of pull out CO2 directly out of the atmosphere to stop that global warming.
Yeah, I think, Michael, that makes a lot of sense. And maybe to even provide a little more specificity, right? When we think about, you know, how do we begin to address the release and accumulation of carbon dioxide in the atmosphere, as Michael was saying, there are solutions for carbon-free renewable energy production, right? We have solar, we have wind, we have these other ways to get energy, which is of course critical to us as a human civilization. But most of the things around us every day that we use are coming from those petroleum resources. So I think the real special feature of this story is this ability to kind of build with biology, right? It's to be able to take carbon from the atmosphere and use biology to make stuff. And actually, you know, biology is really great at making stuff. And so if we can imagine strategies like the one we reported, where we can use microbes to help decarbonize sectors, we think this could have a really important impact.
And actually, you talked about some of the challenges in the chemical industry. I think using those gases, like, for example, synthesis gas, which is carbon monoxide, carbon dioxide, hydrogen, is something that the chemical industry does for 100 years. It's the Fischer-Tropsch process. But the problem is it needs to have a very, very set ratio of the gas. The gas needs to be highly pure, and you work with sort of high temperatures. I think biology can do essentially the same things at kind of ambient temperatures, and in particular kind of is very tolerant to impurities, and can really work with very chaotic gases. So if you look at a steel mill, even there you have a very chaotic fluctuating gas stream of the waste gas. But if you then look at something like municipal solid waste that can be turned into a syngas, I think depending on what ends a certain day in a landfill, I think you get very different gas compositions, and the biology is flexible enough to handle this, whereas I think traditional catalysts have challenges with that. So biology is very good at taking a very chaotic input and make it very specifically into one product, which then again could serve for other purposes. I think acetone you could make into plexiglass. I think ethanol, which I think is another product LanzaTech produces. We've developed the processes to convert that into jet fuel, into PET, and so on.
Yeah.
So how did you decide how to sort of approach or tackle this problem when you were first sort of looking at your options?
Yeah, I think I mean for us when LanzaTech started, I think the ambition was certainly that using waste feedstocks, and then what feedstocks are today abundantly available and not used, or only used for purposes which they're better options. For example, with some of those gases you can make energy, but it's very poorly efficient, and I think now there's different options for that. And then I think we identified that there are autotrophic bacteria which can actually feed on those gases, like carbon oxide, like it's carbon monoxide, carbon dioxide, and hydrogen. That, for example, yes, steel mill gases are kind of have those composition. In fact, I think there's even speculation that life on Earth started with kind of those C1 gases with CO2 down at those hydrothermal vents in the deep sea, where I think you have those kind of gases, reduced compositions, and also some metal centers which are found in the enzymes of those bacteria as well. But the key challenge was, I think, you need to then supply those gases to the microbes in an efficient way. And I think 10, 15 years ago, there wasn't any genetic tools for those microbes available.
Right, yeah, that makes sense. I think, yeah, if we could go more into detail in the acetone and the isopropanol. What made you choose those two as a target, and why? And I guess, what were the initial barriers that you had to overcome? What was the first steps in getting this process started?
Yeah, for one, there's obviously a market for those molecules. I think acetone is kind of used for plexiglass, isopropanol actually in hand sanitizers, and as they're both solvents, I think isopropanol can also be turned to polypropylene. But they also have this very similar characteristics to ethanol, which I think is, from a process perspective, is an advantage. So you can imagine we can just change the microbe, but then use the same plant infrastructure that we have. And so I think that was certainly a motivation as well. And there were pathways known to make acetone and isopropanol. So to our knowledge, there's no organism in nature that can convert those gases to acetone or isopropanol, but there's some sugar fermenting microbes that can turn sugar into small amounts of acetone. So we had a starting point where we could get the genes from this pathway. When we first started, I think we put in genes to make acetone into this organism. We only produced very small amounts. So I think the challenge was in identifying the right sets of enzymes, and then also optimize the host to really kind of channel all the flux into those products.
Yeah, and I think this latter step, this is one of these areas where the tools from synthetic biology really accelerated our ability to develop strains that could manufacture commercially relevant rates and titers of, in particular, acetone and isopropanol. And, as Michael said, we started by putting in the end, like, let's imagine we take the acetone story as a starting point, right? So it's 3 steps, 4 different enzymes that are used for that. We mined these enzymes from a historical collection of Clostridia strains and others that had been used in manufacturing different types of products in Clostridia over and through the 1900s, called the David Jones collection. And after we got some better sets of enzymes, we still only saw a little amount of acetone produced. And one of the wrinkles, of course, when you try to engineer heterologous pathways in living organisms, is that oftentimes you get byproduct or shunt pathways that are actually pulling the carbon away from, in this case, the acetone molecule you wished to make, but to another product. In this case, we saw 3-hydroxybutyrate, for example, as a side product that was being made. And so what we had to do was find out which sets of enzymes in native metabolism of the organism were responsible for these side products, and then delete them. And that actually in and of itself is a big challenge, because in Clostridia, it's slow growing, there are transformation idiosyncrasies and others. And so actually trying all of the computationally predicted deleterious enzymes could have taken us, months to even years of time. And so one of the things we ended up doing was implementing a cell-free system, a way in which we take kind of the biochemistry of cells, but use it without a living organism to accelerate the process. And this allowed us to actually identify the deleterious enzymes in a reasonably short period of time, just let's say weeks instead of months to years. And that allowed us to then prepare an engineered organism that had a high selectivity of, let's say, acetone production, and ultimately could be transformed to process optimization steps at LanzaTech for scale up and maintaining kind of stable and high rates for weeks at a time.
Wow. So, okay, let me make sure I've got that right. So you did the pathway optimization was cell-free, and that's how you kind of got the enzymes correct.
Yeah, one of the ways for kind of implementing the strain optimization strategy was that we just used a very simple cell-free approach that enabled us to identify candidate genes for genomic knockouts. And that was able to be done in just some very simple experiments that took just a couple of weeks. Whereas if you kind of would have iteratively tried to knock those enzymes out from native metabolism in the host organism, it would have taken some time, kind of in the months to longer range. We didn't go that approach, so I can't tell you exactly how long it would have taken, but it was exciting to be able to accelerate the design of the process because of this. And I think, you know, overall, this is really kind of pointing to how the tools of synthetic biology are really beginning to help us, you know, as engineers, to think through how to reprogram organisms for social good faster than we ever had before.
And in terms of sort of scaling up the production of these organisms, in comparison to traditional methods, how does your sort of procedure compare?
So I think one quite different aspect from traditional sugar fermentation is that we have to sparge the gas to the bacteria continuously. So you can essentially think about a fermentation process, but where we have the gas kind of bubbling through the fermenter, and I think we feed that gas constantly to the microbes. But since we have to feed the substrate constantly, I think this allows us then to also develop a continuous process. So I think now we can essentially feed the gas into the bioreactor along with some nutrients, I think some salts and metals. And the bacteria essentially eat the gas and I think make acetone or make other products that we've engineered the microbe for. And at the same time also divide. So I think, and then at the back end, we take off the product, the acetone or the ethanol, and essentially can return some of the water, replenish some of the nutrients, and have the gas essentially sparking through there all the time. But I think it's been also a kind of engineering challenge, of course, with those gases. I think some of those gases like carbon monoxide, hydrogen are not very soluble in liquids, in water. So I think it's been kind of developing bioreactor technology, which does that at a low energy efficiency and is scalable. So I think right now LanzaTech is operating 2 commercial units with 500,000 liter bioreactors. And essentially we're doing this at the moment for a strain that's essentially a non-modified strain that's naturally able to make ethanol from those gases. But I think now we can of course bring in the strains that make acetone and isopropanol, and really the framework which Mike also highlighted. I think we believe that can be applied then to make other chemicals of interest, and also even to other organisms and accelerate that, which I think is very exciting. I think really that interdisciplinary approach there, where I think obviously the engineering to scale up the process. I think the tools we've developed to kind of accelerate the strain engineering with addressing some of the difficulties working with this organism. The life cycle analysis, I think we've developed a lot of omics technologies that we used also to optimize the process, where we partnered with Oak Ridge National Lab. And Mike mentioned the sequencing of genomes there, which I think we've sequenced over 300 strains from a historic strain collection. Some that go back to kind of Chaim Weizmann, which actually was at the University of Manchester in, I think, start of the last century. So I think it's really a lot of these disciplines coming together, which enabled this work. And I think also the private-public interface there that kind of came together, and I think also enabled, for example, through funding from the Department of Energy then to make this work happen.
So you mentioned the life cycle analysis. So firstly, what is a life cycle analysis, and how did you decide on sort of the criteria for the life cycle analysis?
So yeah, I think the life cycle analysis essentially looks at the carbon footprint of a process or of a product. And I think for us, I think that was really important to do in this context. I think we wanted to benchmark how it compares our process with the traditional petrochemical process that uses fossil feedstocks. And of course, I think it's important that we kind of have a process that is cost competitive, which I think we've developed this process to rates of 3 grams per liter per hour and high selectivities of over 90%, which makes this commercially viable. But then I think we also have an advantage over the traditional manufacturing in that we can show our process is actually sustainable and we can have an achieved carbon negative process. And essentially the life cycle analysis looks at all the inputs of a process and essentially what energy the process has and what emissions are associated with that. And then essentially looks at what's the footprint of a product that's produced considering all of those steps.
That are there sort of are there objective standards that sort of you judge you judge your product against, or is that something determined between sort of the sort of more sort of, let's say, the public and the private kind of interface, the researchers and the like sort of the industry?
Yeah, I think as I mentioned, I think the motivation for making acetone and isopropanol, certainly there is the market, we had a pathway, but I think then we also looked at what's the economics of a process, where do we need to get to, how does that benchmark against the traditional process, which I think the life cycle is an extension of that as well. And we certainly use that, and I think we've employed some genome scale modeling and some kinetic modeling, which I think really allowed us to then identify what's the limits of the process, what can we get, and then feed that into the economic models. Really, I think to in the first place make sure we work on something that's relevant, that's feasible, and where we can make an impact, and we actually can kind of evaluate that for several other molecules, and then I think pick the molecules that we think we can make the biggest impact.
Yeah, in terms of the life cycle analysis itself, also it relies on, data from the pilot scale model coupled to industrial scale possibilities. So it is important. You asked the question, is this connected to kind of know-how at the industrial level, and it does rely on kind of background information from those processes already, but as Michael said, and what's so exciting is basically the result of that analysis shows us that we turn a process that today when we make these types of chemicals, we do so in a way where we release carbon dioxide into the atmosphere in the overall process, and now we've inverted that, right, where we're able to take carbon dioxide from the atmosphere to lock it into these products.
Yeah, that's great. Could you go into like brief detail into this? So in your paper, it's sort of split into 3 steps of the pathway optimization and the strain optimization and the process optimization. Could you explain what exactly you did at each step in that optimization process?
Yeah, I think we started out, of course, I think looking for a pathway. I think in the first step, I think we then had this acetone pathway, which I think is essentially 3 catalytic steps catalyzed by 4 enzymes. And essentially what we did, I think we pulled out variants for each of those enzymatic steps. So I think we've sequenced this collection of 300 strains that were used historically for making acetone, butanol, and ethanol in a mixed fermentation from sugars, I think start of the last century. I think in that case, that process was phased out of because of costs, and I think because of acetone, the selectivity to acetone was poor. But I think there's been 4 decades of development to optimize the process for making those products. So I think rather than just relying on sequences from natural isolates, I think we looked at in the collection, is there some genes that are different from those environmental genes? And in fact, I think we've identified over 50 unique genes. So I think we had essentially 10 enzymes for each of those steps, and we just randomly put those into different combinations with different promoters and looked at, can we find combinations that lead to more acetone production, and which I think we were successful to do. But even then, I think we still only had maybe 5% selectivity to acetone. So I think a lot of the products still got diverted to ethanol as a main product, and to 3-hydroxybutyrate, as Mike described earlier. So I think we used essentially those cell-free tools and also the omics tools and modeling to really optimize the flux towards acetone and isopropanol, optimize that to a selectivity of over 90%.
Wow!
And
so you went from 5% to 90%.
Yeah, which I think also shows how powerful those tools are.
Yeah. How did you use the modeling and the omics?
Yeah, I think once we had a kind of eliminated some of the first 5 products, I think we've got up to selectivities of 60-70%. But then I think in the kind of omics analysis with proteomics, we identified there are some enzymes which I think we would have expected to be expressed higher. And the modeling also pointed towards those enzymes could potentially be a bottleneck. So I think what we then did is adding a second copy of those enzymes, and with that we could kind of eliminate that bottleneck and really open up the pathway and get to improve the flux to those 90%. And before we went into the cell to do that, we again used the cell-free system where we could just increase the concentration of that enzyme and could see the positive effect as well.
Right. Yeah.
Once we had then a strain that was working that well, I think we optimized the process to achieve those higher rates of over 3 grams per liter per hour, and went from the lab scale to a 150-liter pilot reactor that's been in field at the site we have in the US and Georgia. And that reactor resembles then more the commercial reactor.
Going from the lab to that large scale, there must be some quite a lot of the way you say it in like 2 sentences, but I feel like is it quite a lot of work, or is it not that? Is it quite easy to do?
It is. I think we had the advantage, of course, we could build on our experience from scaling up the ethanol process. So I think there we've had over 100,000 operating hours at that kind of a pilot demonstration scale before we could go to a commercial unit. So I think there was a lot of experience we could tap into from that perspective. But it is a big step going from the lab, from a kind of a benchtop continuous stir tank reactor to kind of a reactor that more resembles the commercial operation. I think that's where the process is actually operated, not by scientists, but by operators with commercial or with chemical grade, that's industrial grade chemicals, with not as pure gases. So I think that's been a big step, and showing also the stability of the process there for weeks at a time.
Yeah, that's cool.
I'm really curious about sort of your paper sort of hints at this kind of idea that the process you have created can be used for lots of different different chemicals. What is it about your process that can be used for different chemicals? What is it like? What's modular about it? How can it?
Yeah,
how does it? Yeah.
Yes, Sarah, it's a great question about kind of the modularity. And in this case, the key modular piece is actually the microbe. It's actually the engineered biological system itself, in the sense that LanzaTech has developed a process in which they use a microbe that produces ethanol. And so, you know, in kind of theory, the dream, right, is to now swap that microbe out with a new one that's been programmed, as we showed in this case, to produce acetone, or a separate microbe that's been programmed to produce isopropanol. Or you could actually imagine, and I think one of the things that we're really excited about is the idea to make a wide array of different chemicals sustainably in a carbon negative approach using this platform, right? Just by changing the enzymatic systems that are present in the microbe that make different types of chemical products. And so the flexibility therefore comes from using different engineered microbes. And in some sense, you could imagine that this would allow one to flexibly produce different products, even based on market needs. You know, because you could swap out different microbes in the process if it could be designed to be so. That's, you know, there's a lot of work to put into scaling the process, but in terms of the modularity, the key modular piece is the microbe. Michael, I don't know if you have other things to expand on. But.
No, I think we're very excited about the opportunities here. I think the platform, as Mike mentioned, I think is there for making ethanol. I think the platform itself has a unique flexibility in terms of feedstocks the process can use. So I think our commercial unit right now using steel mill gases or waste gases from the ferroalloy process. So I think essentially waste gases from heavy industry that are otherwise not used or able to make power at low efficiency. But the process can also utilize any kind of biomass. So for example, agriculture waste or actually urban waste, municipal solid waste, you can gasify into a syngas, which again is carbon monoxide, carbon dioxide, hydrogen, which can feed into the process. And then these organisms are actually also able to use CO2 with, for example, hydrogen or renewable electricity. So there's a lot of flexibility on the input side. And now we can, with synthetic biology, enable a lot of flexibility on the output side, where I think initially we started with ethanol, but we can now be building up now to have acetone, isopropanol, but there's a lot of other opportunities. And on a proof of concept level, we've already shown that we can modify the organism to make over 100 different molecules. And I think the framework we developed here, we hope that with that we can accelerate development of some of those other products as well.
Have you already got an idea of what other products you want to specifically focus on next?
Yes, I think as LanzaTech, for example, we have a couple of projects with partners and customers. So for example, we work with BASF, which is a big chemical company, to make octanol. So I think it also shows the interest there from the chemical industry. But then also we have partnerships with, for example, Lululemon to make kind of monoethylene glycol, which again shows also the interest there from brands. So I think it's really kind of looking at where do we have partners that are interested in molecules, and then which molecules are kind of may make sense from a kind of metabolic perspective and market needs, to bringing that together. So I think at the moment we have several kind of products in development.
That's great to hear. That's very exciting. So you did, so the Clostridium species that you use specifically, that was from the David Jones collection, did you say? Or
no, I think the Clostridium that we use is called Clostridium autoethonogenum. So I think that's essentially been isolated 30 years ago by a Belgian research group, and then deposited in a strain collection. And I think there's been then 20-25 years no paper on this organism. And essentially we started out kind of looking at a whole range of different autotrophic organisms that have been deposited in strain collections. And I think this organism was in particular interesting, since it could make ethanol. So I think we've started to develop the process around this, and then obviously develop the genetic tools for this organism as well.
Yeah.
But the
Yeah, in terms of the David.
No, go ahead.
Sorry,
Mike.
Michael. I was just to say, you had asked about the David Jones collection. That was it's a resource, a collection of strains that we had sequenced with JGI and the Department of Energy to gain access to sequences that encoded enzyme function, for example, for acetone biosynthesis or others. So
Right.
Just to make that clear.
Yeah, I thought, I don't know why, but when I read that, I thought it was very interesting to see, because you said it was from, work from the beginning of last century, and it's just cool to see how people are building on things that are 100 years old and getting this new technology from some work.
Yeah, no, it's exciting. I think it was one of the first processes where it's been shown we can actually make chemicals with the fermentation process, and actually the process is really kind of has a very remarkable historic function in that I think Chaim Weizmann was first to develop this process. I think later on became the first president of Israel because of this achievement in this process. I think during the war, I think acetone was required to make cordite as a kind of shooting powder, and I think was really kind of critical for the Allies to win the wars. I think that process had quite some significance there. But then with once cheap petrochemical oil became available, I think that process was phased out because I think the yields were not great. I think the sugar feedstocks got more and more expensive. It was only kind of carried out in politically isolated countries, for example, in South Africa. And David Jones was a researcher there in South Africa, which worked with one of the last of those ABE fermentation plants, and I think saved this collection of strains, which I think spans 4 decades of development, which I think he then took to New Zealand, where he started a lab there, and we were fortunate enough to have access to those strains. I think initially we hoped any of those strains may be able to grow on gases, which I think unfortunately wasn't the case. But now with the sequence technology advancing that much, you're able to really kind of sequence that whole collection, all 300 strains, and I think pull out the genetic information, which we then use to modify this gas fermenting Clostridium that we have.
Awesome. Yeah, I think that's all the questions I have. What about you, Sarah?
Yeah, same here.
Yeah, that was great. I think we'll wrap it up there then.